I'm going to be telling you about several projects whose overall goal is to establish human stem cell systems that can capture multiple Angelman syndrome genotypes and epigenotypes. And we're hoping to be able to address pretty comprehensively all of the different forms of Angelman. So my lab is at North Carolina State University in Raleigh. And before jumping in, I just wanted to introduce you to the team because these are the young scientists that are actually pushing forward the work in the lab. This was pre-pandemic last year, and when restrictions lifted, we immediately ran into the lab and then also ran to the Halloween Cow Creamery to feed our brains with some sugar so we can get stuff done in the lab. But you know, I just want to really point out and introduce them because they're really the ones getting the work done in the lab, but also it's just very inspiring for myself and encouraging for me to work with them because they're very passionate about bringing benefit and just making the world better in the future. So I think they're going to be the future leaders generating a lot of the new ideas and approaches in the next several years. So there's really two goals that I wanted to achieve today. I had kind of two missions. So the first mission is to share with you what we are doing, what's their overall goal, and what's the overall strategy. But the second mission is, like Allison said, information is power. And so I know that some of the science and the details can be kind of eye-glazing. So what I'm going to try to do, though, is really share with you what we are actually trying to do at the molecular level. And the reason for this is because a lot of what we are doing is molecularly trying to change the DNA, the genome. And so I think it's important to understand what exactly we're trying to do there. And to do that, I'm going to be using this schematic over and over again. But I'm going to try to stick with this scheme so that you can kind of see what's changing, what's not changing. And this is a version of the two chromosomes, so one copy from the father and the mother, that you've seen in some of the other talks. And what I've done is tried to focus in on a few regions. So there's a lot of text and a lot of boxes, but really just try to help you focus in on a key core regions. So this is the region, this is UBE3A, and these are all the surrounding genes nearby it. And this is in a neurotypical genome where you have UBE3A on the maternal, the mother's copy, and this is turned on. So I represent that by this light blue. So all if the gene is blue, then it's turned on. And on the paternal allele, we know about this imprinting effect where we have the long non-coding RNA UBE3A ATS, and that is transcribing and inhibiting the paternal copy of UBE3A. So to indicate that this copy is off, I've made it red. Okay, so in a neurotypical case, in neurons, we have one copy of UBE3A that's turned on. And much of what we know about Angelman syndrome and what a lot of the therapeutic developments have been focused on understanding what happens when the maternal copy of UBE3A is deleted or it's gone. And so this could be either through a mutation in UBE3A that ablates the protein being able to be produced, or it could be through a large deletion. In this case, this is a class 2 deletion in which the maternal copy is gone. And so when this happens, you only have one copy of UBE3A, but that copy is shut off. And so you have no UBE3A. And so studying the absence of UBE3A, the deletion of UBE3A, has been done through mouse models and through human stem cell systems, and they have been used to look at, to identify therapeutics. One of the key approaches is stopping the stop that we've talked about, where you can have ASOs or CRISPRs to inhibit the long non-coding RNA, and that allows the paternal copy of UBE3A to then reactivate. Okay, so I just wanted to go through all of that again just to reorient you using kind of this particular schematic because they all look a little bit different. Now the issue is that Angelman syndrome is diverse. right? So there are many different genotypes and epigenotypes. So class 1 and 2 deletions, there are over 30 known UB3A mutations that have either loss of function, gain of function, or functions that we still don't really understand. Uniparental disomy, imprinting center defects, and also, as Dr. Keung mentioned, there are other genes that have been deleted as well, and we don't understand what those do entirely, and also we don't know whether they're druggable or not. So what I'm going to do is kind of walk you through each of those different genotypes, because those are essentially what we're trying to recreate in a human stem cell system. So this now is a version of a large class 2 deletion where the deletion includes these 4 genes as well. And so you've actually deleted many genes on the maternal allele. So one of the questions that we'd like to develop stem cell systems to be able to answer is, well, what are the roles of those genes? What are their impacts on neuronal function? And can we maybe develop systems where we can rapidly rescue and express those genes and understand what happens? There's many UB3A mutations, so I'm representing that by this red vertical line. So one of the questions is, can we develop models that have all of these different mutations, which is very difficult to do because it can take a long time to generate each mutation? Can we develop a platform which allows us to generate many, many mutations very quickly and create all of these models efficiently? The other question, and the other reason why it's important to generate these models, is that if we think about the therapies like stopping the stop, do we understand how those therapies will act in a case where you have UB3A mutations rather than a deletion? So for example, if you stop the stop here, that would reactivate the paternal UBE3A, but would it also increase or affect the expression level of a mutated version of UBE3A as well? And that might be something that could be good, but it could also be bad. And then finally, there's also epigenotypes, so uniparental disomy and imprinting center defects, of which this is representing 2 paternal chromosomes that have been inherited. And in this case, and also in the case of imprinting center defects, what you end up having is these long non-coding RNAs that are being expressed from both chromosomes. So both the maternal and the paternal. And what that means is that you actually get UB3A on both UB3A copies are actually present, but both of them are silenced. And so you have no UB3A being expressed. And so one of the questions here is then, well, if we're going to stop the stop, how does that affect UB3A levels? In this case, if you stop the stop, you might be stopping the stop on both chromosomes, and you might have double the amount of UB3A than that you would expect, and that might not be something that we'd want. And so can we develop model systems to screen therapeutics, maybe be able to tune the level of inhibition and reactivation of UB3A? Okay, so those are basically the molecular genotypes that we'd like to be able to recapitulate in human stem cells. So Angelman syndrome is very diverse. I did want to take a broad view just to note that there are multiple approaches to try to model these genotypes that I think are all going to be important. So Dr. Keung actually mentioned, for example, the development of mouse models to reflect large deletions. But you can also potentially create mouse models or rat models or other animal models to reflect all of these different genotypes. I think this is very important. Dr. Keung also mentioned previously in the previous talk about deriving human-induced pluripotent stem cells, where you take cells from each genotype and you can create stem cell lines to reflect those genotypes. What I'm going to tell you about today is a parallel approach where we are going to, instead of starting from many different cells, from many different people, what we want to do is start from one stem cell line. It has one genetic background. And we're going to use molecular engineering and CRISPR tools to edit the DNA so that they now reflect the specific molecular DNA and genotypes within the UB3A region of each of these different AS genotypes and epigenotypes. we also want to be able to develop basically cassettes or kind of plug-and-play regions in the genome where other researchers can efficiently include and express other genes that are lost in Angelman and basically be able to understand what those are doing. Now, the reason why, the key distinction between this approach versus iPSCs, the induced pluripotent stem cells, is that all of these cells are what are called isogenic, meaning everything else in the genome is the same. All of the genetic sequence is the same except for the Angelman mutations or deletions. And the reason why this is important, so I've represented this by everything in the cells is gray, so all of the cells are gray except for the Angelman change, which is one of these different colors. And so this is isogenic, and the reason why this is important and potentially useful is that it can reduce the variability in experiments and in therapeutic screening. So because all of the cells have the same genetic background, you don't have variability that's introduced from all of the tens of thousands of other genes that are present in the genome. It can also help facilitate, therefore, comparisons between researchers, between institutes, between companies. If we're very careful about it, and which we are, we should make sure that we're working with stem cell lines that are not restricted by the federal regulations, and so that we can leverage fast funding to have a multiplier effect to help us leverage NIH and NSF funds to also perform this type of work. And we're engineering these to be shareable, so we want to generate these cell lines so that they will reflect AS genotypes, but also will be customizable by other researchers in a very easy way. So what are we actually doing? So when we are making one of those colored stem cell lines, what we want to do is start with these stem cells and do some sort of molecular engineering to convert them to have an Angelman genotype. And so what we're doing here is we're starting with a neurotypical stem cell line, and we're using CRISPR, which are basically complexes of proteins and RNAs that can bind very specific regions in the genome. And so you can program where it goes and binds in the genome. And so we're going to use these to go and bind this site, okay, right here and right here. And you can link different functionalities to the CRISPR. So the natural function of CRISPR actually is to cleave, to cut DNA. So we can cut DNA and then reseal from here to here to create a large deletion. You can also fuse to these CRISPR molecules. You can drag it to this region, for example, and edit the epigenetic state. And you can recreate the UPD or ICD states. So that's another project that we're tackling right now. Now, using CRISPR is pretty inefficient. It's something like 0.01% efficiency. And so it's doable, but we have to do a lot of selection and screening. And so the analogy I like to think of about this is that my wife says that I don't cook very well. But if I have a recipe, I'm OK. So what we want to do is create a stem cell line that basically it provides a recipe for other researchers. So what we're doing is using something called a landing pad. We can use CRISPR to knock in this small piece of DNA with relatively decent efficiency. And this landing pad has these brown sequences here that we'll call kind of like flip-in sites. And what these brown regions in this landing pad allow us to do is, with high efficiency, flip in large cassettes of genes that can include 3, 4, multiple of these genes that are lost, for example, in class 1 or class 2 deletions. We can also flip in UBE3A mutants or UBE3A wild type into these regions with higher efficiency. And so our goal is to create this line, be able to share it widely, as well as with the recipe for how to knock in all of these other all these other genes and genetic material as well. The other advantage that these landing pads offer is that we can bring in some tools from synthetic biology to help us as well. So for example, this is a landing pad that we designed and knocked into human embryonic stem cells. And this landing pad has a unique feature in that we can dose it with a small molecule, an antibiotic called doxycycline, and it can actually turn on the expression level of a gene. So you can imagine if this red fluorescent protein was UBE3A, we would be able to titrate and tune the amount of UBE3A that's present. And this could allow us to understand UPD as well as deletion situations. There was also a question previously about SNO RNAs and SNORD 115, 116, other genes that are in that intervening region. You could also use this system to understand the effect of aberrant regulation of those genes as well. So that's the engineering aspect. That's how we create the stem cell lines. Now the question is, what do you then do with them, right? So there's several things you can do with them, but in our group, what we're interested in is differentiating them into these human cerebral organoids, which are these microtissues. The penny here is for size, but they contain 30 to 50 different neuronal cells from the human brain. So that's one of their powers, is that they contain a lot of cellular diversity and they better mimic the brain, the completeness of the complexity of the brain. They also exhibit a lot of structure. So for example, cortical lamina that resemble the outer layers of your brain. And so you can also get some structural information as well from these models. And so we've generated these organoids before from stem cells that mimic, or stem cells that have Angelman genotypes, in this case a class 2 deletion, and you can see that they exhibit the silencing of UBE3A, so there's no UBE3A in the neuron, but there is UBE3A in other cell types that surround it. So it mimics the specific silencing and imprinting of UBE3A within neurons. And because it does that, we can also use these organoids to do therapeutic screening. So this is just an example of topotecan or indotecan reactivation of paternal UBE3A. So the signal here in red is UBE3A, and when you add indotecan in these organoids, you can see reactivation of UBE3A. You can also look at functional changes in the neurons in these organoids, where this is an AS organoid, and this is an AS organoid treated with indotecan, and you can see it rescues it to kind of functional patterns similar to that in a neurotypical organoid. Okay, and then lastly, when we are working with organoids and stem cells, I just want to mention one of the real advantages of this model system is that they contain all of these different cell types. Right? They have multiple different types of neurons from all different regions of the brain, typically this would be a problem. It'd be something that's not good for experiment. But a lot of advances in analytical technologies now have allowed us to really start taking advantage of this complexity. So this, for example, is where we've taken an organoid, broken it up into single cells, and each dot represents a single cell, and we're able to identify the gene expression signatures from each individual cell and identify what cell type it is. And so the colors represent different cell types within these organoids. And what's really powerful about this is then now we can start to ask questions like, if you have a therapeutic, does it impact UBE3A and gene expression differently in different cell types? So for example, in this type of excitatory neuron versus an inhibitory neuron versus a progenitor cell, we can get much more specific about what brain regions, what cell types, and how therapeutics differentially act in different regions of the brain. So with that, I'll just summarize that the overall goal of our projects are to create stem cells that can capture these AS genotypes and epigenotypes, and to generate organoids out of them to understand the effects of these genes, as well as see whether we can establish screening protocols. We've assembled a team of really passionate scientists and students. Begum and Chris over there are here today, and so if you have questions about their work, I'm sure they'd be very eager to tell you more about it. And with that, I'd just like to thank past and present members of the group that have been pushing these projects forward. And of course, FAST for funding this work, that we hope to report some new reagents and tools for researchers and companies in the coming days. And I'd just like to mention also that Stormy Chamberlain, Dr. Keung, they've already shared cell lines with us. It's a very collaborative research community and something that I very much appreciate. And if you have any questions, feel free to drop me a line.